When we started the Human Genome Project, a lot of people said that we're not going to get those difficult regions. But 10 years ago, the technology started to advance, and it allowed us to get more and more of the genome. I became kind of, maybe the right word is obsessed with finishing it. And in working with a wonderful group of young people, around 2020, we were able to actually figure out how to sequence one human genome from beginning to end.
When I first started working at the University of Washington, we were sequencing little tiny bits of DNA, 100 at a time. And now we're generating 1,000 times that on a daily basis.
And we use the human genome as a way to predict unstable regions.
Is that too long? Those too long? 30
Yeah.
And then I tested children that had unexplained developmental disabilities. And we discovered about a dozen new genetic causes of intellectual disability, autism, and epilepsy doing that type of work. For many families, actually understanding why their child has this problem is actually very, very, very satisfying for them. It gives them a reason why. The power of that has been connecting those families with a common genetic cause. And I'm using genomics and sequencing to do that.
I think research is honestly one of the best jobs ever. You know, as basic scientists, we are at that forefront of medicine.
And there's some hints, not proofs yet, that we can explain more of the genetics of autism by completely understanding these difficult regions. You have to unpack the complexity if you're going to come up with solutions. And we believe by actually building a genome that represents all humans, that will have more power to find disease association from collaborations around the world. And in so doing, we will help everybody, people from African descent, from Asian descent, from Melanesian descent, because those discoveries will contribute to new drugs that will actually be used to treat people all around the world.
The stuff that we do in this lab, while it may not directly impact a patient tomorrow, right, like what a surgeon would do, reality is that 5 years from now, the discoveries we make in this small space will directly impact patients.
This is what we set out to do 30 years ago, and we're now at the point where we actually can do it. We just hope that we can continue to do that work, because when people are going to be tested for risk for cancer, for risk for disease, you're going to need the complete sequence information. One human genome is not enough. 1,000 is probably not enough. And we're at 300 right now. And we're moving forward to get to 500 next year. And it will benefit everybody if we do it right.